And a very warm welcome to everyone, and thank you for joining today's edition of the Precision Nanosystems Teatime Webinar series. It's my pleasure to introduce today's talk and speaker. Today's talk will be entitled Cationic Lipids Offer New Possibilities in Liposome and LNP Engineering for RNA Therapeutics, and it will be presented by Guéguen of Polyplus-transfection located at Strasbourg in France. So just before we start, just a little bit of housekeeping. So first to introduce the PNI team. So I'm Richard, I'm the team leader for Europe. Also work very closely with my colleagues Guéguen and Suha, who are our two regional sales managers based in Berlin and London respectively. And also a key part of the team are our field application scientists looking after scientific, technical and applications related matters based across the continent. That's Ed, Martin and Sarah. Thank you once again, everybody, for joining us. I'm sure we'll have a very interesting presentation today. And now it's my pleasure to hand over to Claire, our speaker today. Thank you.
All right. First, I would like to thank PNI for giving me the opportunity to present our data today. So today I'm going to talk how cationic lipids offer new possibilities in liposomes and LNP engineering for RNA therapeutics. So let's start with a quick introduction on Polyplus-transfection. So at Polyplus, we produce transfection reagent for since now than 20 years. So we offer transfection reagent for different applications, for cell and gene therapy, with the example of production of lentivirus AAV, for also in vivo and DNA RNA therapeutics, for antibody and recombinant protein discovery, and also for life science research and also drug discovery. Our headquarters, R&D and manufacturing facilities are based in Strasbourg in France, and we also have support and commercial teams in Europe, Americas, Asia and China. We offer also GMP grade reagent for some applications, so for example for virus production and also in vivo DNA therapeutics. And we are also certified since 2002 in ISO 90001. So that's, as I said, at Polyplus, we have now more than 20 years of expertise in DNA and RNA deliveries. We first launched in 2001 in vivo jet-PI, which is product for DNA delivery. And 3 years later, we launched a GMP grade of this product. We also have transfection reagent for siRNA delivery with interferon, and also for RNA delivery for in vitro application with jet messenger, and for in vivo application with in vivo jet-RNA and in vivo jet-RNA plus. That it's a new formulation that we're gonna launch this year, and we're currently working on cationic lipids for LNPs, and we are planning to offer a GMP grade of these cationic lipids. So we have several method tools for non-viral delivery. So we have cationic polymers, but also cationic lipids. So in vivo jet-PI, which is a cationic lipids, can be used for DNA, mRNA, or other RNA deliveries. It's mainly used for proof of concept drug discovery, but as I said, we have also GMP grade version, so it can be used in therapeutics for clinical trials. And it's ready to use reagents, so you just have to mix the polymers with your nucleic acids and inject into the patient or into the animals. We also have cationic lipids with lipid-based reagents, so with in vivo jet-RNA, which is a liposomes. It can be used for mainly mRNA delivery. It's also mainly used in proof of concept discovery. It is also ready to use reagents, so you just have to mix the liposomes with your RNA. And we are working also on 2 other methods. So we are working on cationic lipids for LNPs. So will be also used for mRNA, DNA, or other RNA delivery, and it will be a component of LNPs, and it could be used in a therapeutical application. And the last one is hybrid polymerization. So we are currently also working on that in the R&D chemistry team, also for mRNA, DNA deliveries, and for therapeutics also. So in this talk, I'm just going to focus on cationic lipids used in liposomes and also in LNPs. So in non-viral nucleic acid delivery, we face different challenges. The first one, we want to have an efficient and safe distribution. So the choice of the molecule you want to use for nucleic acid delivery is quite important. For example, with lipids, if you use cationic versus ionizable lipids, you want to have the same biodistribution. The second one is we want also to have easy product to use, to handle, and to supply. The easiest example is when you use liposome versus LNPs. It's quite easy to use liposomes, whereas with LNPs, you need a microfluidic equipment to formulate your LNPs. The third one is we want also to reduce the cost from the research to clinical trials to markets. So the ideal is to have a complete package of molecules that you can use from research to clinical trials. And the last one is quality and freedom to operate. So you want to have a GMP product and also the freedom to operate, especially as we've seen now with LNPs, the lipids, you don't want to pay royalties to use the lipids to formulate your LNPs. So the first generation of cationic lipids used in LNPs were DOTMA and DOTAT. So they were really efficient. They are still really efficient to deliver nucleic acids via LNPs formulation, but they can be toxic because they have trimethyl ammonium parts, which can induce toxicity. So the alternative is to use ionizable lipids, as we've seen with different vaccines currently on the market. So we have the D-Lin MC3 DMA ionizable lipids used on On-Patrol, but we also have the two other ionizable lipids used in the COVID vaccine 19 with Moderna and BioNTech, which are really efficient, but sometimes they can be a bit limited on the biodistribution with these ionizable lipids. So at Polyplus, we've been working already since 15 years now on lipids, on cationic lipids. And so here it's our discovery process. So we start with the synthesis of cationic lipid libraries. We do different kind of characterization. We check that we can scale up the production of these lipids. We identify and check the purity of lipids. Then we do a screening of formulation. So for that, we also use the nanoassembler Ignite from PNI to do different formulation of lipids with a different method, different buffer. We then check that the physical physical study of our LNP lipids or LNP formulation. So we check the size, the potential of our particles. We obviously we check the encapsulation efficiency of our lipidic formulation. And then at the end, we check obviously the transfection efficacy with in vitro and in vivo transfection. And we also do a toxicity study. So here it's our proprietary cationic lipids, so which is quite different from the first generation of lipids. So it's composed of a hydrophobic tail with a trident shape. We also have a linker for covalent bonds, or we can also make biodegradable molecules. And we have, which is quite different from the classical cationic lipids, we have a polar head, which here it's imidazolium with a positively charge. and we have a functional group which hides also the positively charged polar head, and that's why that functional group can reduce the toxicity of our cationic lipids. So at Polyplus, we have two methods to use our cationic lipids. So we have mRNA liposomes, which are off-the-shelf products. So we just mix our lipidic formulation, so jet messenger or in vivo jet RNA plus containing our cationic lipids. We mix this formulation with mRNA, and the mRNA is then encapsulated inside the liposomes, as we can see here, which can be a bit different from lipoplex, where we have more the mRNA on the surface of the lipoplex, and which represents poor encapsulation efficiency, whereas with our liposomes, we have all the mRNA which is encapsulated. And we also formulate LNPs using microfluidic technology. And so we use the nanoassembler Ignites, and we mix mRNA with different lipids, including cationic, proprietary cationic lipids. I'm going to start first with liposomes, with in vivo jet RNA. So our first aim is to check that we can use high concentration of mRNA with our liposomes. So we've seen with current COVID vaccine, some of the vaccine were around 100 microgram per ml. So here we can see, we look at the size of our liposomes mixed with mRNA for up to 1 hour at room temperature. We tested different concentration of mRNA, and we can see we managed to go up to 300 microgram of mRNA per ml, and we can see we have a liposome which are stable for 1 hour at room temperature, and we can see that the size it's around or less than 200 nanometers, which is a really good size for in vivo injection. We also check that we can use our mRNA with different size, our liposome with different size of mRNA. It means that our liposome is a quite versatile use. So we tested different size. So we tested a small mRNA around 1 kb, a medium around 4.5 kb, and also an sRNA. And we can see that we have stable liposomes still up to 1 hour at room temperature, and our size is around 80 or less than 1 nanometers. One of the challenges also with mRNA and lipidic formulation, it's also the stability of our liposomes. So we then check also that we can keep for several days our liposome mixed with mRNA. So we first look at the size of our particles when they mix together, when they were stored. So at room temperature or at 4 degrees, and we could see here that up to 7 days our liposomes mixed with mRNA are stable, and we see the size doesn't change over the time, and is still less than 100 nanometers after 7 days at room temperature and 4 degrees. Obviously, we check also the efficacy of our liposomes. So it's a study ongoing. So we've data on this for up to 3 days, where we can see, so we have in yellow at 4 degrees or in blue at room temperature. So our liposomes are still efficient when we store them up to 3 days, even at room temperature. And we can see we have really good transfection efficiency, and the transfection efficiency doesn't decrease. So here we use a luciferase mRNA. So it means that our liposomes are really protecting mRNA from degradation even at room temperature. So we then looked at the expression in vivo. So we did different routes of injection. We first check with IP injection using again a luciferase mRNA. So we look at the luciferase expression here per organs. So we can see by using our liposomes in vivo RNA plus mixed with mRNA, we can target a lot of different organs, especially if you want to do vaccination purpose. These liposomes able to target spleen and also lymph nodes. We can be critical for vaccination purpose. We also did an IV injection. And we can see also via IV injection, our liposomes can target different organs, and it mainly targets lung and spleen. And we can see also expression in the liver, in the kidney, heart, and pancreas. And we also did intramuscular injection, and we also had a really good luciferase in the muscle where we injected the liposomes. The advantage also by using liposomes, we can do dose escalation. We can do dose escalation, so you can just mix a different amount of mRNA with liposomes. So here we've shown that we've used from 5 to 20 micrograms of mRNA via IV injections. And we can see in all the different organs, we have a nice dose response relationships with our liposomes. So we can see in some organs, for example, in the spleen, when you add from 5 to 20, you can see we have a 2-log increase of luciferase expression, which is quite easy to make with our liposomes. And finally, we're also checking that our liposome don't induce toxicity. So we looked at the pro-inflammatory cytokine. We injected 2 different quantity of mRNA via IV injection. Our positive control was LPS. So we can see we have high secretion of different inflammatory cytokine with LPS. LPS secrete a lot of IL-6, IL-12, TNF-alpha, TNF-alpha, and also interferon gamma. And with negative control and with our 2 different quantity of mRNA mixed with liposome in vivo RNA plus, we don't see at all secretion of pro-inflammatory cytokine, which is really good, which shows that our liposome don't induce toxicity in the mouse. Then we've studied the use of cationic versus ionizable lipids in mRNA formulation. As we shown previously that we can use our cationic lipids in liposomes, we also have been working on different formulation of LNPs, still by using the nanoassembler Ignites from PNI. We use the standard lipids that are used for LNPs. So we have helper lipids, which is the phospholipids. We also have a structural lipids, which is the cholesterol. We have a stealth lipid with the pegylated lipids. And we also we tested without and with an additional ionizable lipids. So we first compare our cationic LNPs made with our cationic lipids versus ionizable lipids, so the Delin MC3 from Onpattro vaccine. So as expected, when we look at the charge of our formulation, so in blue we have our liposomes in vivo RNA plus mixed with mRNA. In yellow, it's our LNPs made with our cationic lipids, and in dark blue, it's the one with ionizable lipids. So expected with our by using our cationic lipids, we have formulations which are charged positively as expected, which is different from using ionizable lipids where we have quite a neutral or slightly negatively charged formulation. And this difference in the charge will give us the different biodistribution in vivo. When we look at the size, for liposomes or LNPs, we are roughly have the same size. We are under 100 nanometers. And we also check the encapsulation efficiency. And as you can see here, so we have really good encapsulation efficiency for LNPs using cationic lipids, but also for our liposome, where we can see that all the mRNA is encapsulated in our liposome. And so our encapsulation efficiency is as high as lipids, LNP using ionizable lipids. We then check first the transfection in vitro. So we've tested in different cell line, on adherent cell line first. So we've tested on colon, hepatocyte, lung, but also epithelial cells. So in blue, you still have liposomes in vivo RNA plus mixed with mRNA. We also made LNPs with our cationic lipids, and we tested as a control Moderna and BioNTech formulation. So we've again used mRNA, luciferase mRNA. So here we can look at the luciferase expression in the different cell lines of the 24 hours of transfection. And so we can see with our liposomes or our LNPs, we have really good transfection efficiency. We can be as high as Moderna or BioNTech efficiency, or sometimes can be even much higher. We can induce really a higher transfection efficiency with our lipoplex and our liposome with in vivo RNA, and also with our cationic lipids present in our LNP. We also looked at transfection in hard to transfect cells to see also if in vivo we will also be able to reach and to transfect this kind of cells. So we first looked at transfection of human T cells. So of a positive control, we have our liposomes. So we use jet messenger, which is for in vitro transfection, but we also checked with our liposome in vivo RNA, and we can see here we reach really high transfection efficiency. Around 80% of the cells were expressing GFP. And with our LNP, we can see also we managed to have high transfection efficiency. And when we modulate the LNP formulation, we managed to reduce the size of LNP from 80 nanometers to 40 nanometers. And we can see that we managed to increase the transfection efficiency with smaller LNPs. We then looked also at the transfection of hematopoietic stem cells. As a positive control, we had our jet messenger, which is for in vitro transfection, and we see around 40% of the cell transfected. And with our LNPs, we managed to have higher transfection efficiency, and managed to around 75% of the cells positive for GFP expression. We then look in vivo expression. So we first did an IV injection by using again our liposomes in vivo RNA plus versus our LNPs using our cationic lipids in yellow, and we compare to LNPs with ionizable lipids, Delin MC3 DMA from Onpattro vaccines. And as expected, as we have different charge of LNPs, so with the Delin MC3, we have really high expression of luciferase in the liver and lower expression in the lung and in the spleen. Whereas with our liposomes or with our LNPs using cationic lipids, we have high expression in the lung and in the spleen compared to Delin MC3, but lower expression in the liver. We also compare our cationic LNPs with Moderna, and we had roughly the same profile, still higher expression in the liver. It was roughly the same in the spleen, but still higher also expression in the lung compared to Moderna LNPs, so with our LNPs containing cationic lipids. We also checked that different organs to see that we can reach also via IV or intramuscular injections, that we could be also efficient. So here we did retroviral injection with our LNPs. So as we already seen with our liposomes, our cationic LNPs mainly targets lung and spleen, but we still have expression in the liver, kidneys, heart, and pancreas. And when we also do intramuscular injection, we have a nice luciferase expression also in the muscle where the injection was injected. Finally, we look as we look also as the stability of our liposome, we look at the stability also of our LNPs. So this is a study also ongoing. So we stored our LNPs at 4 degrees without addition of cryoprotector. And we can see here, we compare to our liposomes, and we kept our LNPs up to 13 weeks. And we can see that the transfection efficiency of Cap2 cells in vitro were still really high. And we can see that we didn't see an impact. Our LNPs are quite stable up to 3 months at 4 degrees. So to conclude, we've shown here that cationic lipids are suitable for nucleic acid deliveries. Here I've shown you one cationic lipids, but we also have a library of the different cationic lipids. And our cationic lipids can be used in 2 different methods. The first one, which is quite easy to use, it's a liposomes with in vivo RNA+. We can be used mainly for mRNA, but also for other RNA and CRISPR's delivery. It's off-the-shelf product, so it's easy to use and can be stored so far up to 7 days at room temperature of 4 degrees. And in vivo, it's via IV injection. It mainly targets lung and spleen. But we can also use our cationic lipids in LNPs, and we can use it for mRNA, but also, I didn't show data, but it can be also used for DNA and siRNA and delivery. So far, it's stable up to 13 weeks at 4 degrees. And when we inject it via IV routes, it's mainly also targeting lungs and spleen. Some advantage also with our cationic lipids, we are planning to offer also a GMP grade of this lipid this year and the next few months. And we also have freedom to operate and to use these lipids. So our final aim at Polyplus is to offer to our clients to use the same molecules. So here's the same cationic lipids through the process from R&D to clinical trials. So first in vitro, they can use jet messenger. And then for in vivo proof of concept, they can use in vivo RNA. And after they can choose between the 2 options, they can still keep using liposomes, so they can still keep using in vivo RNA+, which is under development for also the GMP version. And if they want to use LNPs, they can use the cationic lipid, which is present in jet messenger, but also in vivo RNA, and they can use that cationic lipids to make LNPs for their use in preclinical or clinical studies. So finally, I just would like to thank people from Polyplus who has worked on this different projects, especially from the R&D chemistry and life science team who made all the data. And I also would like to thank the Mass Clinical Institute where we do all the in vivo experiments. And thank you all for the attention.
So all that remains for me to do is say thank you once again to Claire from Polyplus for a really interesting presentation today, and thank you for everyone for participating and joining us as well. We hope you found it as interesting as I did, and yeah, please do join us in another month's time for the next Teatime webinar. Thank you all very much.